What's up, Hippie Biopagans? Today we do 2015 number 4 on circadian rhythm and evolution. Many species have circadian rhythms that exhibit an approximately 24-hour cycle. Circadian rhythms are controlled by both genetics and environmental conditions, including light. Researchers investigated the effect of light on mouse behavior by running a wheel with motion sensor to record activity on actograms as shown in figure 1. So as you see here, we have this new diagram you've probably never seen before. It's showing us that when the mouse is inactive, we just have empty space. And when the mouse is active, it's shading in with this kind of dark bar. For the investigation, adult male mice were individually housed in cages in a soundproof room at 25 degrees Celsius. Each mouse was provided with adequate food, water, bedding material, and a running wheel. The mice were exposed to daily periods of 12 hours of light and 12 hours of dark for 14 days, and their activity was continuously monitored, and the activity data is shown in figure 2. So when they had light, we see that the mouse is inactive, but in the darkness, the mouse is active. After 14 days in L12D12, the mice were placed in continuous darkness, DD, and their activity on the running wheel was recorded as before. The activity data under DD are shown in figure 3. So as you see that when it's continuously dark, we do have a little bit of a shifting and how much they spend in that darkness or in that running varies based on the mouse. So part A, the nervous system plays a role in coordinating the observed activity pattern of the mice in response to light-dark stimuli. Describe one role of each of the following anatomical structures in responding to light-dark stimuli. So to be honest with you, the animal systems are no longer in the curriculum, but I did want to talk about this because this kind of looked at signal transduction and it looks at communication. So photoreceptor in the retina of the eye, this is going to be where we're going to receive the signal. And so the point of the photoreceptor is going to be to receive the signal and then send that message to the brain. The brain is going to be responsible for kind of integrating and determining what the response is, and the motor neuron is how we're going to send the message back out to that structure. So detecting the light-dark stimulus and initiating a signal, brain integrating and processing coordinating information, and then the motor neuron transmits the signal from the brain to an effector. So a student says photoreceptor in the retina of the eye plays a role in sensing the light in the mouse's environment. It responds to the lack of light, and so when it is dark, the photoreceptor sends a signal to the brain, indicating that it is dark. The brain receives the signal from the photoreceptor, telling it that it's dark, processes the signal, and then sends the signal to the appropriate neuron to act. In this case, it is the motor neuron. The motor neuron receives the signal from the brain to move and in turn causes the mouse to begin movement activities such as using the running wheel. Part B is based on an analysis of the data in figure 2. Describe the activity pattern of the mice during the light and dark periods in the L12D12 cycle. So this is just asking us to look at this diagram and tell us, what do we see? Well, I see that when it is light, that the mouse is not active, but in the darkness, I do see it's active. It spends 12 hours inactive and then 12 hours active. So you can say that it's active only during the dark periods and it's inactive during the light periods. You can say it's only active in the dark or it's inactive only during the light. Students said during the dark periods, the mouse is almost constantly active using the running wheel. During the light periods, the mouse rarely uses the running wheel. So they, of course, use that first bullet point. Part C, the researcher claims that genetically controlled circadian rhythm in mice does not follow a 24-hour cycle. Describe one difference between the daily pattern of activity under the L12D12 conditions and under the DD conditions, and use the data to support the researcher's claim. So the claim was that it's not just genetics. So if it was just genetics, then I would expect to see the exact same thing happening in my L12D12 as I do in the DD, if it was completely under genetic control. But because of the fact that I don't see that it stays in that same pattern, I see that the amount of time shortens in one or the other. It doesn't exactly spend exactly 12 hours at any given point. It's a little bit more broken up. It has to do with some type of environmental condition. So the description is that it's active begins a little earlier each day. The active inactive period is shorter than 12 hours a day. The daily circadian rhythm is less than 24 hours, or the pattern of activity is going to shift each day. All of that information can be given just looking at your diagram. The second part says support. So it says you use that data to support the claim. So without light, the active and inactive periods are determined only by genetically controlled circadian rhythm. And if it was a 24-hour circadian rhythm, the pattern of DD would be the same as the pattern that we saw in the L12D12. So under L12D12 conditions, the mouse is constantly active for 12 consecutive hours when it is dark, and then shows inactivity for 12 hours that it is light. When looking at the mouse placed in the DD conditions after 14 days of L12D12 conditions, it first continues the same pattern of activity. However, the days increase, the mouse deviates from this pattern. By the 40th day, the mouse now exhibits about 6 hours of activity, followed by roughly 9 hours of inactivity. This combines to a 15-hour cycle, differing from the 24-hour cycle in the L12D12 conditions. This shows that without the light stimulus, the mouse is genetically set to a different circadian rhythm cycle. Part D says investigate the claim that exposure to light overrides the genetically controlled circadian rhythm. The researcher plans to repeat the experiment with mutant mice lacking a gene that controls the circadian rhythm, predict the observed activity pattern of the mutant mice under L12D12 and under DD conditions that would support the claim that light overrides the genetically controlled circadian rhythm. So we have a mouse that no longer has the genetic control. So if there was any type of genetic control, like that's not actually going to work anymore. So I would expect to see that mutant is going to have a normal rhythm. It's still going to look like the L12D12 because the light is what's controlling it. Versus when we looked at the mutant, there's going to be no pattern. There's no way that we're seeing a certain sequence that's occurring within that 24-hour cycle because there's no genetically controlled rhythm, and it requires the light in order to determine whether it's going to be active or inactive. So then be constantly active or constantly inactive. So the student says, lacking the genetically controlled circadian rhythm, the mice will be active only when it is dark and inactive when exposed to light. Under L12D12 conditions, the mice will be inactive for 12 hours during the light and active for the following 12 hours during the dark. Under DD conditions, it will be constantly active as it is constantly dark. Eventually, the mice will probably die from exhaustion and overexertion. Part E, in nature, mice are potential prey for some predatory fish that hunt during the day. Describe two features of a model that represent how the predator-prey relationship between birds and mice maybe resulted in the evolution of the observed pattern of mice. So here we're seeing that the predatory birds, they hunt during the day. So if the mouse is active during the day, then logically it's going to be eaten. So those mice that are able to be out and about when it's dark are going to be less likely to be consumed, which we're going to see that since it's under genetic control, those traits will be passed on because they're more likely to survive, more likely to have more offspring, and those offspring, of course, will be more likely to survive. So we'll see that the trait is going to be inherited and more likely to be inherited. Okay. So we have to talk about the selective advantage. Selection for individuals active at night, selection against individuals active during the day. The day active variants are susceptible to predation, or saying that the night active variants are going to avoid that predation. You also have to talk about the reproductive success. So mice selected for are going to produce more offspring, and the mice selected against will produce less offspring. So students said the birds that prey on mice hunt during the daytime when there is light. If the mouse is active during this time, it will be more likely to be killed and eaten by the bird. This could have caused the mouse to be active only during the dark in order to avoid being preyed upon to survive, reproduce, and pass on its fit genes to the next generation. The mice without the genes for inactivity during light would have been killed by the birds, preventing them from passing their genes to the next generation. Over time, this has caused all or most mice to have this gene, and it is the most favorable for survival. This is known as survival of the fittest, or natural selection. So I hope that was helpful. Remember, AP Bio Paying was just a test bio.